Hello, ladies and gentlemen, and welcome to today's broadcast on high throughput native MS with robust ion source operation for the analysis of proteins and protein complexes by Dr. Caroline Chu, an LCMS application scientist with Agilent. And I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Before we begin, I want to encourage you to engage with us. You can submit as many questions as you want at any time you want during the presentation. Just simply type them into the Ask a Question box to the left and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just click on that support tab found at the top right and use the Ask a Question box on the left. Please join me now in welcoming Dr. Chu. You may now begin your presentation.
Hi, thanks for joining me today as we talk about a little bit on high throughput native MS with robust ion source operation for the analysis of proteins and protein complexes. As a refresher, just for those that are not familiar with native mass spec, native mass spec is a little bit different from denaturing mass spec. In native mass spec, the protein retains its native form in its folded form under biological conditions at pH 7, usually with the ammonium acetate as our buffer, ranging from 50 millimolar to 200 millimolar concentration. It is a charge limiting application, so it's a little bit more challenging to ionize. You can do it two ways, essentially online size exclusion chromatography for online desalting and offline buffer exchange, and then introducing it into the mass spec with usually nano ESI. So denaturing conditions is more commonly used. It's a low pH and an organic buffer is usually used, which causes the protein to unfold and you get a very nice charge envelope. It is definitely not charge limiting, and it's usually done by reverse phase and can be used by any one of our ionization sources such as the AJS, ESI, and nano ESI. So in my previous webinars, we talked about comprehensive approaches to doing native mass spec. Here we're just looking at the liquid chromatography introduction of these proteins under biological pH 7. So here we're using a 1290 Infinity 2 LC UHPLC, and you can do it one of three ways actually. The traditional way is by doing it with nano ESI. So here on the top, you can see the nano adapter. So that converts the high flow to nanoflow, and you can interchange it with the G1992A nanospray interface. Or alternatively, we've done a lot of cap flow work with our standard ESI source. You can do it under cap or microflow rates. And what we'll talk about today is something we've recently introduced earlier this year, which is the Agilent jet stream source under standard flow conditions. So any one of those conditions I outlined in the previous slide with the nano ESI or AJS source can be coupled to any one of our high resolution accurate mass QTOF systems. So what I'm showing you here are some of the commonly used platforms for this analysis. So here you have the 6230 TOF from the left, a 6530 QTOF, a 6545 XT, which will be the primary focus of our talk today, as well as the 6560 ion mobility QTOF. So as you can see, there's different options you can use for the high resolution accurate mass system. You can still do it with our base model TOF or the base model QTOF. For larger complexes, you'll see the 6545 XT offers you an extended mass range. And for those higher order structure analysis, where you get that added dimension of ion mobility separation for conformational studies, that's where the 6560 would be a great instrument to utilize. So how did this high flow work come about? So this is work done with my colleague, Pat Perkins, and he basically got inspired by doing a rapid online buffer exchange system. So we initially had a lot of capillary flow work And then now we've moved on to high flow work based on this publication by Vicky Wysocki's group. And so we decided to utilize that kind of publication as a platform and migrating it over using our Agilent JetStream technology with our mass specs. So here you can see the publication on the left and an application note that kind of covers what the Agilent JetStream is about. So from that, we basically was able to generate a poster for ASMS 2020 this year. It's MP499. And so what we'll do in this talk is kind of walk through this poster, go into a little bit more details about how we use standard flow native MS for these protein complexes. So these are the analytical conditions that we use for the Agilent JetStream source. For those who are not familiar with the Agilent JetStream source, this is basically what it looks like. So on our mass specs, all our sources are interchangeable with each other. So whether you have a TOF, QTOF, or a triple quad, those sources are all interchangeable between each of those platforms. And so here we utilize the Agilent Advanced Bio SEC cartridge guard column, and we basically leave it at room temperature, and our injection volumes vary. We have a 20 microliter loop on our 1290 Infinity 2 system. So we can inject upwards of 20 microliters, depending on the concentration of your sample. Mobile phases range from 100 to 200 millimolar ammonium acetate, and the flow rate is a lot higher than what we normally would use, which is 0.4 mils per minute. And it's an isocratic run, and essentially it's a 6-minute run. Below are the source conditions outlined for the AJS source. And what I've gone ahead and done is kind of give you a brief introduction to what the Agilent JetStream source is. So you can find more information on the application note that I had just shown previously. And so basically the Agilent JetStream source, it basically kind of gives you a way of culminating the spray. So here is what it would look like without the superheated sheath gas that is introduced. So you see here, your sample is in green. The solvent is in the blue droplets. The drying gas here is in the black ion. And then the orange would be the Agilent JetStream gas, which is introduced with the AJS source. So here it is without the Agilent JetStream gas. So you can see where the spray is more wider, and you can see, more of your sample along with the solvent does get entered into the mass spec itself. So you have an increase in noise. And in here you can see, this is a picture from the Agilent JetStream application note where you can see the spray starting at 25 degrees C. So when you enable the superheated sheath gas with the Agilent JetStream source, you can now see the sheath gas being introduced here, which now kind of focuses in the spray a lot better, where the neutrals are then moved out down into the waste, and it focuses in the samples into the mass spec itself and eliminating a lot of neutrals from going into the mass spec. So now you've essentially improved a lot of your signal versus the noise that's entering the mass spec. And then here is just an illustration of what the Agilent JetStream source looks like at 350 degrees C. So with the superheated sheath gas. So now you can see the spray is a lot more focused in. For the analytical setup, it's basically you just put in the Advanced Bio SEC guard column here. You can see that here. And we can use it with a 10-port valve plumbed to emulate a 6-port valve, or you can just have a 6-port valve. And essentially, you can use the valve changing procedure so that you can do some offline cleanup and desalting. To give you an idea of what the chromatogram looks like, here's a picture of the total ion chromatogram using alcohol dehydrogenase, ADH. So alcohol dehydrogenase is like a 146 kilodalton tetramer, homotetramer. And so that was our base standard for doing a lot of the method optimization and development for this whole procedure here. And so here on the left, you can see the TIC of the alcohol dehydrogenase tetramer. And you can see where the highlighted region is where the ADH does elute off the column. And then followed by this larger peak, which is mainly small molecular weight singly charged species. And to give you an idea of what the mass spectra looks like, you can see on the right is the mass spectrum of ADH, and you can see the charge envelope. It's far more extended into the 5,000 to 6,000 MRZ range, and it does extend out further with the AJS source. So for the source optimization, not only did we take into account the temperature of the gas, the drying gas temperature, we also took into account the sheath gas temperature, the different flow rates, as well as the nebulizer pressure. And so for both of these, you know, if you're familiar with some of our previous work on native MS, we did a lot of work with the CapFlow with the microflow nebulizer. So here we're using the standard flow nebulizer with the AJS source, but it is still possible to use the microflow nebulizer with the AJS source, and you'll see an improvement in your sensitivity. And so what you'll see here is just monitoring a single charge state, MRZ 6147, the area of that, that charge state versus the sheath gas. So changing in the sheath gas and then the changing in the area of that single charge state. So you can see the plot here on the left, which is a 24 plus charge state. And then if you were to also monitor the response of a couple different charge states, kind of like the apex of the charge envelope with the nebulizer pressure, you can also see the response of, you know, the shifting of the charge envelope along with the nebulizer pressure here. So once all the initial experiments were done with the source optimization with alcohol dehydrogenase, a number of standards were explored under these analytical conditions to ensure that, you know, we have the right conditions for these non-covalent complexes. So more commonly used, you know, we're using monoclonal antibodies on the 6545 XT. And so we use the nismab as well as Herceptin, and we tried those two compounds out. And so here you can see on the left is Herceptin, 3.15 micrograms on column. And so what you'll see is the charge envelope as well as in the inlet here, a zoom in at the apex of the different charge states. And so you can still see the glycoforms retained under these analytical conditions with the native MS. On the right is a nismab with comparable concentration of 3 micrograms on column, the charge envelope, and the zoom in of the apex of the charge envelope. So you can see the relative intensities and the distribution of the charge envelope between the two monoclonal antibodies are very much similar. And so what we did was went ahead and used Unideck, which is a free software platform by Dr. Michael Marty. And it is a great platform for using for native MS because it does help with the deconvolution. And it does have some great features where it does also annotate a lot of the charge envelopes. So it's actually very informative and a great way of generating data. And so we've been using a lot of Dr. Marty's Unideck for a lot of our non-covalent complexes, whether it's nano-ESI, CAP, even the standard flow work. And so what you'll be seeing subsequently in the next couple slides with the standards are the analysis using Unideck for the native MS work. And so what you'll see here on the left are the charge envelopes. And then what you'll see is an overlay of the raw data, which is in black, overlaid to the fit data where there is some peak fitting as well as some smoothing occurring over the charge envelope itself. So this is what it would look like after the fit data overlaid to the raw. And then to the right are the annotated peaks with the charge states. And you can see the distribution across the charge states. And then once deconvoluted, you can see the deconvoluted masses. And then we went ahead and zoomed in so you can see the glycoforms. And so we went ahead and tried a number of other complexes. So for ADH, as our standard, we went ahead and used the smaller proteins just to see, because, native mass spec can be utilized for, a number of different proteins, and they vary in size. So we wanted to kind of go across the mass range a little bit to give you kind of an idea of how the standard flow work may be helpful for doing a lot of the native work in your laboratory. So here we see the raw data. This is off the XT without any special smoothing or peak fitting. And you'll see from the left here is bovine serum, albumin, which is a monomer, at 66 kilodaltons, Oenolase, 93.6 kilodaltons, and it's a dimer. And then our standard for this work, alcohol dehydrogenase at 148 kilodaltons, it's a homotetramer. And so first we have the bovine serum albumin. You can see this is 20 picomoles on column, and with the Unideck deconvolution on the upper left is the fitted data, which is the smoothing and baseline and peak fitting over the raw data. To the right are the annotated charge states for this particular protein. And then what's really great is that there's also a heat map where it's the plot of the charge versus the M/Z. And it may be a little bit more challenging to see here, but you can see that you can see the monomer trend line, as well as there's a dimer formation there, which then coincides with what we see with the deconvoluted results, where we see the monomer at 66 kilodalton. Then we also see a dimer a little bit further up. So it did dimerize in the mass spec. Next, we see Oenolase, which is a dimer itself. So again, the fit data is on the upper left, and then you can see on the upper right are the charge envelope with the annotations. And what's really nice here is that you can actually see which peaks correspond to the monomer and which peaks correspond to the dimer. And so the monomer, you can see the peak annotations in these purple dots. And then what you'll see for the dimer are these red inverted triangles. And then in the heat map, you can now see the two different trend lines for the monomer and the dimer. And then, so once deconvoluted, you can associate the two. So we do see that for some compounds, this higher flow may need some more additional work to keep it in a dimer form, as well as some may just have weaker interactions that may not be amenable to higher flow rates, which doesn't mean that it's not possible to do. We do have alternative options. So here we look at alcohol dehydrogenase deconvoluted. Again, the fit data on the upper left, charge envelope on the right. And now you can see the beautiful trend line for the charge envelope and the deconvoluted mass. So we decided to take it one step further and try to go to the higher masses. So we went ahead and explored pyruvate kinase, which is a 237 kilodalton homotetramer, and then glutamate dehydrogenase at 318 kilodaltons, which is a hexamer, and then beta-galactosidase at 465 kilodaltons, and that's another tetramer. So you can see it is spanning across the mass range, which is one of the benefits of the 6545 XT. That is one of the systems that does go to the extended mass range of 30,000 ml/Z. So here we look at pyruvate kinase. You can see the fit data overlaid onto the raw data itself, and in the annotations of the charge envelopes. And you'll see different types of annotations there, which correspond to the different peaks. And then you can see now the heat map, and then you can see the deconvoluted mass for pyruvate kinase. Again, at 20 picomoles on column. Next, we look at glutamate dehydrogenase, 318 kilodaltons, the hexamer. And again, you can see the fit data overlaid to the raw data on the upper left, and then the charge envelope distributions with annotation. You can see now how it's all plotted nicely down here in the charge versus the M/Z. And you see it deconvolutes well into the 300 kilodalton range. Lastly, we looked at beta-galactosidase, and we're looking at a 465 kilodalton tetramer, again, 20 picomoles on column, and you can now see the charge envelope with the fit data overlaid to the raw data, and then the charge envelope with the charge state annotations. And then you can also see the beautiful heat map here with the charge versus M over Z and a deconvoluted mass itself here. And so, you know, this is work that we have put together this year. And so we've now gone ahead and extended it and applied it to different applications. Here, for example, is a recently published poster that our intern, Kate Simamaker, presented at a recent conference last week. And we're using this exact analytical condition, the standard flow native mass spec, to characterize non-covalent complexes of BSA with hydroxycinnamic acids by native MS. So she's exploring different library of small molecules and looking at different interactions between the BSA and these small molecules. And as I mentioned before, you know, as you've seen some of the examples, it may not all be amenable to the standard flow conditions. It would really depend on the user, and we would definitely want to work with you on getting the right conditions. So, you know, you will see some cases where these complexes or interactions may not be amenable to these standard flow rates. It doesn't mean it's impossible on our systems. So what I'm saying is that you can still explore other options. And so I'm bringing back to this slide here. We can still use electrospray, ESI microflow rates. And so we're looking at, instead of a 6-minute run, you may be looking at a 13 to 15-minute run, depending on how you want to adjust the flow rate. So it's still a very robust and high throughput approach to doing this analysis compared to the standard nanospray approach. However, the nanospray is a little bit more amenable to those where you are sample limited. So you would actually need to do the offline buffer exchange and run it by nano. And so with that, I wanted to also introduce to you the cap flow rates, as I mentioned earlier. So you're not limited to the standard flow. So here, we've used the polyhydroxyethyl-A column. It's a 1x50 millimeter column. So it's a little bit longer than what the standard flow column would be. And you're running essentially pretty much the same kind of buffer analytical conditions, but your run times will be slightly different depending on your flow rate. So here we're running at cap flow rate about 5 microliters a minute. And, you know, you're looking at about a little bit longer time of 30 minutes. However, if you're going at 15 to 30 microliters a minute, you're looking at like about 15 minutes for your runtime. So again, there is an approach, whether it's standard flow or microflow or nano ESI, to do these analytical approaches for native MS. So with that, I'd like to go ahead and summarize what we went ahead and talked about briefly today. I went ahead and introduced you to our standard flow approach with online size exclusion chromatography with the Agilent jet stream source, which is about 6 minutes long. And the data can be readily analyzed using Unideck or with our bioconfirm software. Non-covalent complexes can be readily identified using the advanced bio-LC QTOF. So what I'm showing you here on the left is GrowEL, which is what we've done in the past using nano ESI. It's an 801 kilodalton protein. And what you'll see is the analysis done by Unideck, and you can see the beautiful charge envelope with the charge state annotations, the charge versus M over Z plot, as well as the deconvoluted mass. So again, this method is not just limited to the XT. You can use this method on the 6200 TOFs. You can use it on the 6500 series QTOFs. The XT is more highlighted because it does have the extended mass range and the vacuum, which is more amenable to lower concentrations. And before I conclude, I want to acknowledge that this work was done in collaboration with my colleagues at Agilent, especially Pat Perkins, who has now retired. And so this was a great honor of mine to be able to present with him this year some of the neat work that we're able to do with native mass spec. And with that, I'll go ahead and conclude.
Thank you, Dr. Chu, for that informative presentation. We will now move to the live Q&A portion of our presentation. Just ask a question using the Ask a Question box on the left and click Send. Dr. Chu, it looks like we already have some great questions coming in. Our first question is, how easy would it be to switch from standard flow to capillary flow native MS?
So that's actually a pretty straightforward approach to this, because your system, if you recently purchased your mass spec within the last couple of years, it already comes with an AJS source. And if you're familiar with the Agilent platforms, our sources are interchangeable. So you can easily use the AJS source and try the microflow nebulizer and drop the temperatures down and switch it to, microflow or capflow. Or alternatively, what I had presented was using the ESI source. So you can just switch that over, put the microflow nebulizer, and then use a 1 by 50 millimeter column to do your online SEC for native.
Thank you so much, Dr. Chu. And do you perform any sort of baseline subtraction on your data before deconvolution?
So that's a really great question. So using the Unideck software, it does offer you the ability to do some baseline subtraction. I avoid it if I don't have to do the baseline subtraction. But for some of the proteins, you'll see kind of the baseline kind of rise towards the higher M over Z. So that's where the baseline subtraction, along with the peak picking for the predicting the peak shape, does come in handy for the deconvolution.
Thank you very much. And I want to thank our audience for these great questions coming in. Our next question from our audience is, I have a question. The native MS spectra, the Z envelope of the Herceptin, the NIST, and some of the other proteins did not look like a normal distribution in your slides 12 and 13, which seemed to be a bit different from the native spectra I've seen in my own data and other published data. Do you have any thoughts about that?
That is a great question. We also noticed the same thing when we first initially ran these experiments. And so, we've have a good repository of what the distribution looks like for native MS, because we do have it by capillary flow, microflow, nanoflow, and we've done some CE experiments on it. So what we do suspect is that there is some possible charge stripping that's occurring, which then causes your M over Z mass shift to be on the higher M over Z, which doesn't have as much of a Gaussian distribution, but it tends to kind of tail into the higher M over Z. So we're currently exploring, different ways to kind of see if that is the answer, but we are suspecting there is some level of charge stripping that might be occurring.
Thank you so much. Our next question, in which format do the data have to be saved to use in Unideck? And the audience member also wants you to know that it's Jacinth from Laval University in Quebec, and great presentation she wants you to know.
Thanks, Susie. Hi, Jacinta. Good to see you. The data format that you can use is very straightforward. So basically, you go into your qual or your bioconfirm, and you pick out the spectrum that you're looking to do, and you go ahead and export it as a .csv format. And the easiest way is just kind of deleting the first row and the first column. So you essentially just have your data point, and then so essentially your M over Z, and then your abundance. So those two columns, and it readily can be opened up in Unideck. However, I do know that Unideck is supporting the Agilent files.
Thank you so much. And again, I want to thank our audience. This is great Q&A. Do you think that the source ionization parameters would differ for different proteins and have to be optimized for every experiment, Dr. Chu?
In a sense, yes. What we're giving to you is a kind of a general starting parameter, right? And so oftentimes it may need to be fine-tuned. If you look at some of the proteins that we explored, we explored a subset of standards to kind of gauge how the protein response would be. While these are non-covalent complexes, some do fall apart under the standard flow approach, while some do retain their native form. So again, there is going to be some level of optimization that you would need, but at this point, what we present to you, not only with the standard flow, but all our other analytical conditions for native MS, is a good starting point for you to kind of just tweak. So that way you don't have to start from scratch. And it's at least gets you a jump start. And we're currently exploring, as you can see with a recent poster we presented, exploring it with the ligand complexes. So we're seeing how we would need to adjust from there as well. So that's a great question. But yeah, there is going to be some level of adjustment needed.
Thank you so much. And we have time for a couple more questions. Our next audience member asks, I've never worked with proteins before. Can I do it in the native MS?
Absolutely. So basically, we've given you the analytical conditions, the hardware setup, and how to kind of set everything up. But what I would say is that if you don't need to do native and you can get away with denaturing mass spec and still get your intact mass measurements, then we have already pretty much standardized methods for doing that. When you purchase the XT, it comes with the method for denaturing MS along with the column. Native MS is more so for those that are looking for non-covalent complexes, binding studies, that type of workflows. But if you're just looking for intact protein, you can still just do denaturing mass spec.
Thank you so much. We have time for one more question, Dr. Chu. Does Agilent have nanoflow systems coupled to the MS spectrometer?
Yes. So if you saw a slide that I presented at the beginning as well as at the end of the presentation, just to kind of refresh everyone's memory, is that we do offer what we call the nano adapter. So what that is that basically takes your 1290 high flow UHPLC LC system, and it basically turns it into a nanoflow delivery system. So it kind of essentially splits your flow so that you do have nanoflow, and you couple it with our G1992A nano ESI interface on any one of our QTOFs, then you can have your nano flow setup. So you really don't have to kind of get a full on nanoflow LC system, but you can just use your existing UHPLC and convert that over.
Thank you so much, Dr. Chu. And before I offer you an opportunity to provide some closing remarks, I want to welcome John Sawson from Agilent to add a few extra comments to our audience. John, are you there?
I am. Thank you very much, Carolyn. Great talk. Thanks. I just wanted to offer that quick other comment on the low flow, nano flow capability. Carolyn commented very well on the comment of using the nano adapter. But tomorrow afternoon, we're actually going to have a presentation from Linfeng Wu on the EvoSep low flow LC. And so we've signed a co-marketing agreement with EvoSep with the intent that we can now also offer it's hard to put the words robust and nano all in the same sentence, but EvoSep has done a really nice job of improving robustness on low flow with their cartridge-based systems and method transfer. So you'll get a chance tomorrow afternoon to see Linfeng's presentation using the EvoSep low flow. She's running 0.1 millimeter ID columns for that. Thanks.
Thank you so much, John, and thank you again. And I want to thank our audience. Dr. Chu, would you like to provide our audience with some closing remarks before we go?
Sure. So what I presented today was kind of like a brief introduction to doing standard flow native MS, and it was done showing you the data off the 6545 XT. But I just want to reiterate that if you have an existing, TOF, 6200 TOF, 6500 QTOF 6520, 6530, or even eye mobility, you can readily do native MS.
Thank you so much, Dr. Chu, for your time today and for your important research. And as a reminder to our audience, Questions we did not have time for today and those submitted during on-demand period will be addressed via the email you provided during your time of registration. And I want to thank you again, Dr. Chu, and everyone who participated today. We hope you will check out the exhibit hall and enjoy the rest of the Agilent event today and tomorrow. Take care, stay healthy. Bye-bye for now.